Hey everyone, my name is Daniel Kataulan. I'm one of the senior biotech analysts here at Chardan. It is my pleasure to welcome Lisa Ricciardi, the CEO, and Tony Caggiano, the Chief Medical Officer of Cognition Therapeutics, to this leadership call. We'll be talking about the company and recent and upcoming milestones. Lisa, Tony, great to see you.
Thank you. Thanks for including us.
Of course. For the audience members, if you'd like to ask questions during the session, please use the chat function. With that, I think we'll go ahead and get started. So Lisa, if we could spend a few minutes to introduce yourself and Cognition Therapeutics to the audience.
Absolutely. First thing I would say, Daniel, is we're a public company. I'd like to refer investors to our forward-looking statements, reading disclaimers, etc. Let me begin by saying this. Cognition Therapeutics is the AD company many investors have never heard of, right? We were started in 2007 by founding scientists. They ran the company with grant funding until it became clear that the company needed to access the capital markets for funding to grow the business, and that's when Tony and I came into the company. The focus of Cognition since its inception has been what can prevent neurodegeneration. For example, if you looked at the damage caused by A beta plaques in Alzheimer's disease, what would prevent that? This was the origin of the company, and we now have multiple chemical libraries that have been exploring that mechanism. What I can tell you is a company that started with a vision of two founding scientists is now deep into the clinic with multiple clinical trials and results. And what they verify and validate is that the approach we've taken, which is targeting amyloid beta oligomers, in fact, is a way to slow the loss of cognition.
Got it. Excellent. Thank you. So central to the company's pipeline, of course, is CT1812, small molecule in development for Alzheimer's, DLB, and geographic atrophy. So can you talk about the mechanism of action of CT1812 and maybe also mention how it differs from other treatments, either that are approved or are still in development for Alzheimer's disease?
Tony?
Sure. Yeah. So CT1812 was developed in our labs in Pittsburgh, Pennsylvania. And when the founders started the company, their mission was to develop therapies for Alzheimer's disease. And one of our founders had a keen interest in the toxicities of A beta oligomers. Because at the time, and even more so now, science has really shown that it's the small soluble factors like the oligomers that are really the most toxic species of amyloid. So they developed a screen to look at what molecules from our libraries were protecting neurons from these toxic A beta oligomers. And CT1812 emerged from that screen. Now, what we've learned in the many years since that time is that CT1812 is a ligand for the sigma-2 receptor. And when it's bound to that receptor, it reduces the affinity of A beta oligomers for its receptor, thereby reducing the number of oligomers that bind to neurons and increasing the number of oligomers that come off of neurons. So we believe that's the primary mechanism by which CT1812 protects neurons from degeneration in Alzheimer's disease. Now, you had asked how it is similar or different from other therapies. Obviously, the biggest therapies that have been developed in the last several years are the monoclonal antibodies. So those antibodies have taken the strategy of removing large amyloid from the brain. And now, those large amyloid plaques that exist in Alzheimer's disease somewhat are relatively inert, but what they do is that they seed off the soluble factors of amyloid, like the oligomers, which cause the toxicity, and thereby, when you remove it, slow the progress of the disease. So we've taken an approach coming from the other end. So rather than removing amyloid in mass, we're reducing the affinity of that amyloid to its receptor. So a slightly more traditional pharmaceutical approach of modulating receptors and thereby impacting largely the same biology, but from the receptor end, rather than removing the stimulus being amyloid.
Okay, got it. And in terms of, so there, I see the difference in approaches. The plaque hypothesis, right, that you described for monoclonal antibodies, and oligomer hypothesis, preventing oligomer formation. Can you share some of the evidence that suggests that oligomers are potentially greater disease drivers than plaques?
Yeah, sure. So, I don't think there's a lot of controversy in the field right now. I think the view is generally that the plaques, it's not really separate hypotheses. It's really an evolution of the hypotheses that amyloid clearly is a key part of the disease. And what we've come to understand over the many, last 20 years or so is that it's those small soluble factors which interact with specific receptors on neurons and other cells to cause the toxicity. So over the years, the different proteins that comprise those receptors have been characterized. And then you asked about particular evidence. Most of that comes from both cell and animal looking at the application of oligomers within those systems to see toxicity. And then also, obviously pulling cerebrospinal fluid and measuring oligomers in people with Alzheimer's disease. So I think, again, they're not really separate hypotheses, just really an evolution of how the amyloid is interacting with neurons.
Okay, got it. That makes sense. And I guess maybe along the same lines, given their distinct mechanisms of action, would you then expect CT1812 to have synergistic effect if used in combination with other beta amyloid targeting biologics?
Certainly could. Obviously, we have not done those studies, so we're in no way claiming that it does. But certainly in theory, it could. The amyloid oligomers are present in very low concentrations, but they're very toxic. So while the antibodies have been very successful in removing amyloid from brain, there's still likely low levels of oligomers that remain and can continue to cause degeneration. Indeed, if you look at studies, whether it's lecanemab or donanemab, while it's very nice to see evidence of real slowing of disease progression, obviously the disease still continues to progress. So we would be interested in seeing the effects of CT1812 alone and in combination with these therapies.
Okay, got to understand. And then again, mechanistically, so CT1812 is designed to prevent oligomer formation. So when these monomers are not able to form into oligomers, what happens? How are they cleared from the body?
Well, so just a little correction. So CT1812 doesn't prevent oligomer formation. CT1812 prevents or reduces the binding of these oligomers to their target receptors on neurons. So we don't think that we would change how oligomers are formed either from monomers or coming back from fibrils and protofibrils.
I see. Okay. Yeah. All right. So wanted to switch over and talk about some of the clinical data. You've conducted a number of studies for CT1812 already. Most recently, you reported data from the SHINE trial. But before we talk about SHINE, can you spend a few minutes and talk about some of the earlier data that you've collected that support CT1812 in development for Alzheimer's disease?
I'll cover that and then let Tony jump in. So let's begin with our SNAP trial. Our SNAP trial was a target engagement study. It was the first time we were able to see the drug is doing what we expected it to do. First of all, getting past the blood-brain barrier, so no small accomplishment for an oral therapy, and then engaging the target in the whole A-beta cascade as we had anticipated. So this paper has been out and published. That was completed several years ago. From there, we went on to our SPARK study here. Now, why is this important, the SPARK study? For all of us as we age, our brains shrink. With disease and illness, the brain process of shrinking is accelerated. what we saw in this trial was the ability of our drug to slow that process of brain volume shrinkage, mitigate that process. Another paper on our website that's been published. The sequel trial here, we completed and published sometime last year, early last year. Great, fascinating study, because what we were looking at was using EEGs to do a real-time measurement, if you will, of the return to synaptic health. Broadly speaking, that's what we were after in that trial. Why are synapses important? Well, because they're the link between the neurons. They're critical to learning and memory. And so to be able to see in 4 weeks, Daniel, an improvement in the functioning of synapses was very exciting. So that is also published and on our website. Now, turning briefly to SHINE, I'm going to let Tony take you through lots of the details, but at a high level, what I can tell you is safety and tolerability study, multiple secondary endpoints, 150 patients, 1:1:1 enrollment, 100, 300 milligram of drug and placebo. Let's talk about what happened in the study. First, we looked at several secondary endpoints, the ADAS-Cog11 and the MMSE. And in each case, what you can see is an improvement in the slowing of cognitive decline, 39% captured here. Then turning to the ADAS-Cog13 and the cognitive complex, same thing, important slowing of disease progression on the use of the drug. I believe we included in here the functional assessments as well. No, we did not. We saw with regard to the functional assessments, the activities of daily living and the CGTX, that patients in fact showed improvements towards the end of the 6-month dosing period. So at a high level, we looked at this and said the reduction of oligomers from binding, the mitigation of toxicity is having this positive effect. And by positive effect, I mean a 39% slowing of the disease process. This was a safety and tolerability study, so the drug clearly demonstrated its safety and tolerability. The AEs were mild to moderate, entirely consistent with previous trials. No LFTs at the 100 milligram dose, and where we saw LFT elevations at 300 milligrams, they also returned to normal upon cessation of the drug. Last, we had biomarker findings here, which Tony can elaborate on, significant reductions in NFL. NFL is an important marker of neurodegeneration. And so for us, looking at the clinical data and seeing the underpin of the biomarker movement helped us to say, this is a very terrific positive signal, and upon which we want to base future clinical trial work. No symptomatic ARIA. We've been talking about ARIA for years because it is a component of what you see with the antibody treatment. So I would say taken together, we as a company look at these findings and we say, wow, there is great evidence here that oligomer antagonism has a very important role to play in the treatment of mild to moderate AD. By the way, drugs also being studied in early patients in our START trial, and you and Tony were just talking about combination therapy. In that trial, patients will be able to take lecanemab and CT1812, giving us a shot at real-world evidence for combination therapy. What I would say is that the conclusion of it, we look at one another and say, look, in 6 months, our drug mirrored the impact seen with the antibodies, which is to say a 39% slowing in overall cognitive decline. And this stands up well compared to some of the antibodies. With that, I'd love to have Tony comment on further take for the company from this work.
Yeah, so the goals of this study were to identify a safe and tolerable dose, and then show a clear trend for improving or slowing the loss of cognition across several outcome measures, thereby allowing us to design phase 3 trials for CT1812 and mild to moderate Alzheimer's disease. And indeed, that's exactly what we were able to show. As Lisa mentioned, we had no discontinuations due to AEs in the 100 milligram dose, no elevation of liver enzymes in the 100 milligram dose. And the 100 milligram dose performed nearly identically with the 300 milligram dose across all of our cognitive outcome measures. We identified a very clear space at which we can explore CT1812 in larger and hopefully pivotal trials. Furthermore, we saw clear trends for slowing progression of disease across all of our cognitive functional outcome measures. And this is, exactly what the intent of the study was. If you look at the magnitude of slowing progression, again, as Lisa mentioned, we saw about a 39% slowing in ADAS-Cog 13. And this compares very favorably with the two recently approved antibodies, donanemab and lecanemab, where they were slowing, 25 to 30%. Again, obviously, these are big, large studies where they have approved, received approval. So we're not claiming to be any better or anything compared with those drugs, just merely to state that, these changes are real. Indeed, if you think about a 25 or 30% slowing in a course of, say, a disease course of 10 years, and you're talking about 2 or 3 years difference between when people will be hospitalized or need assisted care. So these are really big changes. We're very happy to see those. So finally, understanding a safe and tolerable dose, having, clear evidence of slowing progression, and then seeing nice evidence of disease modification by reducing with the reduction of NFL and the cerebrospinal fluid from the people treated. We have really good confidence that we now can design appropriately sized phase 3 trial to move towards registration.
Got it. Thank you, guys. Let's unpack there. So let's take a couple steps back and look at some of these items more closely, separately. So in terms of I guess the more general question first, in terms of the enrollment criteria and endpoints for this trial, what are the key similarities or key differences from other Alzheimer's disease trials?
Yeah, largely it's similarities. So all of our outcome measures are very well established measures that are used in many different studies. Right? So obviously the ADAS-Cog has been used in numerous studies, and indeed has been used for registration in the case of acetylcholinesterase inhibitors. If you look recently at they used an outcome measure which combined the ADAS-Cog 11 with one of the functional measures. So these are really well established, recognized, and even mentioned by FDA as one of the ways to assess. And we did that simply because we had no reason to look elsewhere, and to follow a path that's both been followed by other companies, by academia, as well as by the regulatory bodies, specifically the FDA. Just seemed like the right course. So again, these are not different. As you would suspect, right? In proof of concept phase 2 trials, you tend to include a lot of different outcome measures, right? Because you want to see, in addition to what you're looking at primarily, what else may be going on, right? with function and with different domains of cognition. So that's why we include these different measures.
One point I would like to throw in there, Daniel, was we were studying a mild to moderate population, not early stage population. There are not many companies, particularly companies of our size, that go after mild to moderate disease. I feel like the stakes are a lot higher, it's harder. And so that was a point of distinction between ourselves and companies.
Got it. Makes sense. Okay. And in terms of dosing, the doses are 100 and 300 milligrams once daily, oral, for this trial. Are there any requirements in terms of what time of day or with or without food? Anything unique about dosing regimen?
Not really. We used to have some restrictions around taking food. That's all been removed as we've done studies to show that the exposure to patients or how people absorb and get into their system is really not changed at all meaningfully with or without food. We ask folks to take the drug in the morning simply because when they come in for clinic visits, we want to be able to control whether they have had drug or have not had drug, whether we're going to be pulling blood for drug levels and so forth, and when we're going to be doing the cognitive assessments. But as far as for the drug to be effective and, again, hopefully slowing the progression of the disease, it shouldn't matter whether you get morning, day, or
Mm-hmm. Okay, got it. Another comment you made was similarity between the effect seen with the 100 and 300 milligram dose. Can you elaborate on that? Are there any endpoints, that performed better with higher dose? Is there any dose response for certain measures that you looked at?
Yeah. So there are a few hints of dose response in the outcome measures in the MMSE. The 300 performed a little bit better. If you look at the NFL, the data is a little more tight with 300, but overall it's really quite similar between the 100 and 300 milligram dose. The clearest point where we see a dose response is when we look at at adverse events, particularly elevation in liver enzymes, being the AST and the ALT. And there, what you see is, as we've reported before, we get a number of folks that discontinued the study in the 300 milligram dose because of elevated liver enzymes. Again, we had no clinically meaningful elevated liver enzymes in the 100 milligram dose. So this was, again, the entire goal of the study is to begin to differentiate effective dose from safe and tolerable dose. And so here we have a clear spot where we can operate, where we see, near complete effect without the issues of adverse.
Okay, I understand. Yep. And another interesting observation for, I guess, on this slide that's on the screen right now, ADAS-Cog11 and MMSE. So specifically, there seems to be a greater separation between placebo and the treatment arms at 14 weeks compared to 26 weeks. Can you share your thoughts on why you think that is, and what would you expect to happen with longer treatment duration?
Yeah, that obviously is here. So my personal belief is that we have two factors that are contributing to this. As Lisa mentioned, we did the sequel study looking at the effect of CT1812 on synapse activity near real time as we delivered. So what we believe is happening, and as our founders had predicted 15 years ago, is that there'll be an immediate boost of cognitive capability based on the fact that there are intact living neurons and synapses that have oligomers. And when you displace those oligomers, the function of those neurons and synapses returns, nearly immediately. And we were able to show that in a sequel study. Normalize synapse activity within 4 weeks of treatment. So we believe that's the initial boost. Now, what we see in the long run, we believe will be slowing of degeneration, again, reflected by reduced NFL. And we believe there we'll need to study for a long 12 months or 18 months. And there we would predict the separation. But that separation will be like.
Got it. Okay. And in terms of the response and the baseline characteristics for patients, do you have any observations? Have you collected any data, that shows any sort of correlation between, severity of disease and at baseline and the magnitude of benefit?
Yeah, and we put a lot of that data in the poster that we presented at AAIC. Now, only prudent to start with the caveat that with 150 individuals, 3 doses, when we start dividing these things into subcategories, right, the confidence in those observations is dwindling. The nice thing that we saw was that overall, pretty much everybody was moving the same direction. So that was a really nice finding, regardless of male, female, ApoE status, whether carriers, non-carriers, on acetylcholinesterase inhibitors, not on acetylcholinesterase inhibitors, age. People are generally moving in the right direction. Now, the only thing that we saw was that the folks who were a little more severe on the moderate end had numerically a bigger difference. Now, that's largely because the mild folks, as would be expected, even the placebo individuals just don't progress that much over the course of 6 months. So in order to see a difference in the people, you need to study for a much longer period of time. So at this point, we really don't see any particular individuals who might not receive benefit from the drug. Obviously, we'll learn more.
Okay, makes sense. Okay, and another thing that you talked about is the biomarkers and proteomics data in this trial. To kind of bring it back all together for the biomarkers, can you talk about the key ones that you think are particularly important? I think you mentioned NFL. Also, maybe talk about MRI findings as well. And I guess importantly, how do these biomarker findings affect your thinking about your next trial?
Yeah, so as you mentioned, the NFL finding clearly supports the idea that CT1812 can reduce neurodegeneration in the diseased brain. As you see here, individuals that were treated with placebo, there continued to be an elevation in NFL throughout the course of the study, whereas that was largely reduced with either the 300 or the 100 milligram dose following 6 months of treatment. So how it guides our thoughts, I think it really confirms for us that we're able to slow degeneration. And again, it just reinforces that what we need to do is do longer and larger studies.
Got it. And for NFL biomarkers generally, they're not always considered a very reliable biomarker in some indications. How are experts thinking about it in Alzheimer's disease? And do you think there is any potential to file for an accelerated approval based on NFL reduction?
Yeah. So yeah, if you look at the revised guidance that came out from the Alzheimer's Association this year, I think Cliff Jack is the primary author. NFL is listed, as one of the biomarkers of neurodegeneration that's reliable in Alzheimer's disease. Now, obviously, NFL is a general marker of neurodegeneration, so it's not specific to Alzheimer's disease, right? But in this population of individuals who have Alzheimer's disease confirmed by amyloid, it's a pretty good indicator that of neurodegeneration. And in these individuals, we can be quite sure the neurodegeneration is because of Alzheimer's disease. And was there a second part of your question there?
I think you answered. Sorry, the other one is the potential for accelerated approval based on NFL reduction.
Yeah, I don't know. We certainly can pursue that. As folks have looked for accelerated approval, right? Biogen received accelerated approval based on amyloid removal. They had earlier studies that were, really quite large. We'll have to see. Our intended path is to pursue a really traditional approach of, phase studies, see significant changes in cognitive measures to support this. Again, if it turns out to be a good path, we will certainly pursue that rigorously. But right now, we're really looking at a traditional path,
Got it. Okay. All right. And one more question for SHINE trial with regards to safety. So overall, as you mentioned, it looked quite clean, across the cohorts. The one notable concern in the higher dose, it was the elevated liver enzymes. Can you talk more about that? How many or what fraction of patients saw that? And in terms of dynamics, did these appear early on during treatment or later on? Just any additional details you can share.
Sure. Yeah. So again, all of the elevated enzymes occurred in the 300 milligram dose. Again, which was a nice outcome for us. Again, establishing a clear place where we can study the drug further, where we see near full effect without the troublesome issues of elevated enzymes. All these individuals, the enzymes returned to normal upon cessation of drug, and they didn't have any other sequela of serious liver injury, such as elevated bilirubin, flu-like symptoms, or anything that would indicate serious liver injury. So for us, I mean, what that means is it's likely in the future we will be exploring doses below 300 milligrams, including 100 milligrams.
Mm-hmm. Okay. Yeah, that was actually my next question. Given earlier PK and PT data, can you share your thoughts on alternative doses or dosing regimens that you might consider testing out to find an optimal balance of safety and efficacy?
Yeah, so as I just mentioned, most likely our larger, longer trials will be using doses below 300 milligrams. Right now, we have 2 studies being conducted that include a 200 milligram dose. So we have a trial that we call START, which is in early Alzheimer's disease. And Lisa mentioned earlier, this study is the one that's allowing folks to also be on antibody treatments as well. That study includes the 200 milligram dose, and it's treating individuals for 18 months. So from there, we'll get a very good idea about the tolerability of the 200 milligram dose. We're also using 200 milligram dose in our dry AMD study, which we call Magnify, and there we're treating patients for 2 years. So similarly, between those 2 studies, we'll have just excellent data around the 200 milligram dose. So while we haven't designed and started that, I would anticipate that we'd be using doses of 200 milligrams and below.
Mm-hmm. Okay. All right. Let's switch gears and talk about the ongoing phase 2 START trial. So you mentioned that already. Can you summarize again the key differences in design between START and SHINE?
Yeah, sure. So START is designed to enroll folks with mild cognitive impairment, early Alzheimer's. So they're just earlier in the disease process than the folks in the SHINE study, where we're treating mild to moderate Alzheimer's disease. So that's kind of the basic difference between the study. In both studies, we're treating with CT1812 daily. We're following cognitive function throughout the course of the study. Now, as the START study is treating individuals with the earlier disease, it takes them longer to progress. So in order to measure a difference between the treated individuals and the placebo-treated individuals, you need a longer period of time. So there we're treating individuals for 18 months. And then also we're using slightly different outcome measures based on the fact that it's a different end of the disease spectrum. So there, the primary outcome is the CDR sum of boxes, which is what was used for the lecanemab approval. That study also, we're studying 540 individuals, evenly distributed between 2 doses of drug and 1 placebo. And then throughout the course of the 18 months of treatment, in addition to following cognition, there's sub-studies using imaging to follow brain structure and amyloid content. There's biomarker sub-studies pulling both cerebrospinal fluid and plasma to look at changes in biomarkers.
Got it. Okay. And can you provide any commentary on the enrollment progress you see to date?
No, Dana, we haven't given any updates on progress. What we don't want to do is find ourselves having to make a disclosure later on in the year. It's enrolling well. It's a big study. It's a long study. We may choose in the future to say, here's where we are. But right now, consistent with how we reported on SHINE and SHIMMER, we haven't said anything. And those 2 trials we reported when the last patient entered.
Got it. Fair. Okay, and another thing that's different here is that there will be a number of patients who are on combination therapy with Leqembi in START. Do you know, or do you have a target fraction of patients in mind that will be on combination therapy?
Well, the simple answer is no. So it's not designed as a combination therapy study where we were studying the effects of the combined drug. We're allowing folks to be on approved monoclonal antibodies simply because we did not want to have people choosing between the first approved disease-modifying drugs in this phase of the disease and our study. Secondly, we really want to be able to accumulate safety and tolerability of using the two drugs together. Now, should we end up having a significant number of people that are on both antibodies and our drug, then we may be able to really determine if there is a additive effect of the two.
Okay, got it. And this trial is being conducted in collaboration with Alzheimer's Disease Trial Consortium, ACTC. Can you talk about the benefits of this collaboration? How it helps identify patients and recruit studies faster.
So one of the things we were so pleased about when they approached Tony to work together, these are top academic centers. I always say to investors, if someone in your family needed treatment, these are the people you'd want to go to. So high quality academic groups, very experienced PIs, that's what we see. The other thing, Daniel, as we look ahead towards commercialization, I think to myself, when you're launching a drug, where do you want to go first? Top centers of influence. And that's who these people are. Paul Asen and his team, very, very experienced. We consider ourselves very fortunate to work with this group. It's a well-organized network of quality investigators. I don't mean to suggest other investigators don't have quality. That's the wrong way for me to convey it. These are top academic centers, experienced people, and people we want to continue to work with as we build out our program.
Got it. Excellent. And to wrap up the discussion on Alzheimer's, with START trial progressing now, the results from SHINE trial, as well as a number of other trials that you've completed. Can you summarize and kind of talk about what you see as the next steps for CT1812 in Alzheimer's?
Yeah, I'll comment on that briefly, giving Tony some time to talk about Shimmer, because I know that's another important topic. We see this data coming together, preparing Tony ultimately at some point to go to the FDA and talk about what does that program look like. He has been working with some of our consultants to determine what does a phase 3 trial look like, how should that be designed, do we have studies that could also be made into phase 3 studies. All the things that you would think a company does at the end of phase 2 reflects our thinking about how we're working right now. We're considering the phase 3 program and what it looks like. There are no specifics yet, but we're figuring out the optimal design, getting input, looking at what are the costs of the study. All those things are ongoing as a result of what we saw in SHINE on top of this data that you see here, the information from our prior clinical programs.
Got it. Okay. All right. So let's continue on and talk about your DLB program, dementia with Lewy bodies program. So CT1812 is in development for DLB. Can you talk about similarities and differences between Alzheimer's and DLB? Obviously, everyone knows a lot of, or heard a lot about Alzheimer's. DLB is, very, very common, but not nearly talked enough about. And mechanistically, why do you think CT1812 would benefit patients, both with Alzheimer's and DLB?
Yeah, sure. So DLB is largely considered to be a disease of alpha-synuclein. Where the alpha-synuclein oligomers are the pathologic species, similar to where we see amyloid oligomers in Alzheimer's disease. Now, it just so happens that, maybe 50 to 80% of individuals with DLB also have amyloid pathology as well. So years ago, when we had demonstrated in the lab that we could displace the found toxic A-beta, our scientists asked additionally, well, maybe we can displace other toxic oligomers in the brain as well. And the next thing that we thought of course, was alpha-synuclein. So we were able to demonstrate that CT1812 and our other sigma-2 ligands could cause the displacement of toxic alpha-synuclein oligomers as well as the A-beta oligomers. So when we saw that we could displace alpha-synuclein oligomers, we became interested in the alpha-synucleinopathies, Parkinson's disease, DLB, and then thinking about the fact that in DLB, a lot of these individuals also have amyloid pathology. To us, that became a really compelling place to test the disease. I mean, to test the drug in an indication outside of Alzheimer's disease.
Got it. And in terms of the rates of disease progression, are there similarities between Alzheimer's and DLB?
You know, it's hard. I think in general, DLB can be a little more of an aggressive disease. It's similar and different, right? In DLB, people have slightly more diverse symptomology. In addition to the cognitive issues, there's things like hallucinations, sleep disturbances, and motor functions similar to Parkinson's disease. There's fluctuations in the disease. There's GI disturbances, and I think it's a little more of an aggressive, progressive disease.
And one of our colleagues this morning was saying to myself and to Tony, he'd come across a paper showing in DLB patients with a higher load of A-beta oligomers on top of the alpha-synuclein, progression markedly faster. So that's one paper. It's interesting because we look at the concomitancy or co-pathology of those two A-beta oligomers and alpha-synuclein oligomers. So that may be another factor interesting for us to follow. More A-beta oligomers, it's a target we hit, faster progression. Can we have an impact visible sooner in that population? It's an interesting question.
Mm-hmm. Okay, got it. And in terms of the current treatment landscape or development landscape, can you provide some commentary on what's out there for DLB patients?
Yeah, that's a pretty short answer, Daniel, which is just about nothing, right? So these patients get acetylcholinesterase inhibitors, they get drugs, as Tony said, for the GI symptoms, often constipation, drugs for motor failures. Often they show up because they've fallen over and over again and they're in the emergency room. And so you're looking at things that might help the gait disturbances. Quite frankly, there aren't any drugs that have been approved for the treatment of DLBs. There are drugs that go after the symptoms. So as Tony said, the variability in these patients, very agitated one day, other days it's called show timing. They're on, wouldn't know they had any kind of cognitive impairment, very variable. And so sometimes these patients are then dosed with the agitation drugs that you've seen approved recently. It's quite a cocktail. I think quite honestly, a lot of clinicians struggle to clearly identify this population and separate it from others. So that's what makes us very eager to get to the conclusion of this study and look at, could we have an impact for 1.5 million people that get confused, often get treated late, and really don't have good options? We have an oral once-a-day drug. That would be significant to move forward with a phase 3 program. Very excited about this study.
Got it. Okay. So let's talk about that ongoing trial. So you have a phase 2 SHIMMER trial with the expected readout later this year, right? In a few months. Can you talk about this trial and when you do readout, what should we be paying particular attention to?
Yeah, sure. So this study designed very similar to the SHIMMER study. We ended up we announced the last patient randomized into the trial. It's 3 arms, just like SHINE study, 2 of drug and 1 of placebo, 300 milligrams, 100 milligrams of placebo. Individuals are treated for 6 months, and we follow their cognitive function. throughout the course of that 6 months with measures very similar to what we used in SHINE, but more appropriate to DLB. We also have measures of sleep function, measures of fluctuation, measures of motor function, again, because these are particular symptoms in DLB, which are different from Alzheimer's disease. So that data will read out very, very soon.
So Daniel, just to underscore something that we learned after communicating about our SHINE study, safety and tolerability study, one might characterize this as signal finding. Tony just mentioned 3 or 4 main sets of symptoms where we have scales in place to see what we can learn. We want to be really clear with investors. The study at 120 to 130 patients was not scaled up to find a p-value. If it turns out that way, that's great. That's not what we thought going into it. And again, 40 patients per arm, it's unlikely we'll find that. So for us, the richness of this program will lie in what we learn about our ability to slow cognition, as well as have a positive impact on any of these other symptoms.
Okay, it makes sense. And in addition to those observations that you'll be reporting on, I mentioned you'll be collecting some biomarker data as well. What are the key biomarkers to look out for patients with DLB?
Another short conversation, Tony.
Yeah, there really aren't any predictive biomarkers in DLB. Now, one really interesting thing we're doing in this study is we're collecting skin biopsies before and after treatment. So there's an assay available looking at alpha-synuclein accumulation in cutaneous neurons, which seems to be pretty predictive of alpha-synuclein load in the brain. And so we'll be able to use that to look at starting alpha-synuclein levels, whether they're abnormal or not, before the study. We'll also pulling plasma samples so that we can look at amyloid monomer ratio as well as plasma p-tau217, which will give us a very good idea of whether these individuals have abnormal amyloid. So from that, we'll have a good idea of which portion of our individuals had both alpha-synuclein and amyloid pathology. And then, as we talked about earlier, perhaps the drug may be more effective in one of those groups or not.
Okay, got it. And I guess a couple more questions about this program. So first, given the high dose cohort in SHIMMER uses the same 300 milligram dose that was used in SHINE study, do you anticipate to see similar rates of elevated liver enzymes in the study too?
I would anticipate similar. Yeah, we have no reason to believe that individuals with DLB are any more or less susceptible than anybody with Alzheimer's disease, and in that case, anybody without any neurologic pathology at all. So I would anticipate similar.
Mm-hmm. Got it. Makes sense. And in terms of the next programs, the next steps for the DLB program, I understand that will be largely dictated by the upcoming data. But as you're preparing for that, how are you thinking about the next steps?
Well, right now the SHINE data was compelling enough for us that we know that we can design a good powered phase 3 trial where if indeed the drug works as well as it did in that study, we know how to get to approval. We need to wait to see what the effect size and the consistency is in DLB, and then hopefully we'll be able to do the same thing. But again, until we see that, we won't.
Okay, got it. It makes sense. All right, and just a couple more housekeeping questions before we wrap the session up. First, can you summarize what the company's cash position and runway is, and if you can share the assumptions that are going into these calculations?
Yeah, we put in our last Q that we have cash into the second quarter. The assumptions that go into that, I mean, listen, we're a small company. We're rigorous with our cash. We're clearly looking routinely to figure out how is it that we can extend our runway. So that's an active process inside the company.
Got it. Okay. And one last question to kind of bring it all back together for investors. Given your recent data, the upcoming data, the longer term data from the other trials, what do you think investors are still missing about Cognition's story, and what should they be paying attention to in the coming months?
Yeah, I appreciate you asking that question, Daniel. First of all, we just demonstrated with an oral drug the ability to slow disease progression by 39%, mirroring what you saw with monoclonal antibodies with a far simpler regimen. There's room for many drugs in this disease area. However, what we've shown was a really important signal in our study that merits future investment. We'll have more data at CTAD about that trial. So for investors to think carefully about what it was we've shown. That's the first thing. Second thing, big catalyst is in the DLB data. It's months away, and that's an important finding. Our biomarker work, we have a lot of biomarker work that preceded SHINE, and here again, we showed the impact of NFL, strong underpin in that conduct of the trial. What have investors missed? It's hard for me to know that. What I do think is the company may not be well and deeply understood. And so that's why we want to get out this story and continue to tell this story. I do think investors should look at our sources of funding. We've received probably close to $200 million in funding, NIA, NIH, Michael J. Fox, the ADDF. Why do I bring that up? Because it is my belief as the CEO, this company is characterized by the rigor of the leadership, principally Tony executing all these studies, and then our transparency with investors. When we sit down and talk to investors, we make it really clear, the good, the bad, the ugly, if you will. And perhaps it's easy for, you could maybe make a different choice and be less fulsome, if you will, in terms of what's going on. So we've chosen to create a culture in this company that says, do it right, communicate what the findings are, build the trust you have with investors, retail and institutional, fund the programs and move on. And we think that's allowed us to build good relationships with strategics as well as investors. So for investors that don't know us, I hope they do. We feel like we have a very exciting program, an oral drug, the impact we've seen, not in one study, in multiple studies. And yes, larger trials will tell us if it gets across the finish line as a phase 3 program. But this is something we believe deserves investment.
Got it. Well, thank you, Lisa. And thank you, Tony. Really appreciate you finding the time to join me for this discussion. And I look forward to staying in touch. Thank you.
For inviting us. We appreciate it.
Thank you.